Welcome back to Rad Onc Smart Review, RadBio edition.
Hey everyone, we are here for our final session in this radiation biology series. It's been quite a journey, hasn't it?
It really has. And today we're tackling three, well, absolutely crucial signaling pathways. You really need to get these for boards and, frankly, for just understanding tumors day to day. RAS-MAPK, PI3K, mTOR, and HI-1α.
Yeah, these aren't just, lines and arrows in a textbook. When they go wrong, and they often do in cancer, they really drive tumor growth and, importantly for us, make those tumors resistant to radiation.
Exactly. So we'll break down how they work, why they're so central in cancer, how they connect to radioresistance, and touch on how we actually try to target them with drugs.
Yep, get ready. We'll cover the mechanisms, the clinical side, and those high-yield facts you need.
Okay, let's dive in. Before we hit the specific pathways, maybe just a quick refresher. Why are signaling pathways, such a big deal in the first place?
Well, think of cells needing to talk, right?
Right.
They need to respond to their environment. Growth factors, stress, nutrients. Signaling pathways are those communication lines. There are these complex networks, mostly proteins, that take a signal from outside, pass it along inside, usually amplify it, and tell the cell what to do, grow, divide, survive, move, or even die.
Like a really, really intricate relay race inside the cell.
Exactly. Or circuits, maybe. And in cancer, these circuits get rewired. They get stuck in the on position. So you get uncontrolled growth, cells refusing to die, adapting to bad conditions, basically. The hallmarks of being a cancer cell.
And that directly impacts how they handle the stress we throw at them with radiation.
Critically. Absolutely. These altered signals profoundly influence how a tumor cell responds, or doesn't respond, to radiation-induced damage.
Okay, solid foundation. Let's get into the first big one, the RAS-MAPK pathway. Sometimes called the MAP kinase pathway. I usually think of this as the main engine for proliferation. How does it get started?
Typically starts at the cell surface. Signals, usually growth factors, bind to receptor tyrosine kinases, RTKs.
Okay, RTKs. Give us an example. Like
EGFR, the epidermal growth factor receptor, or PDGFR, platelet-derived growth factor receptor, FGFR. Lots of them.
right, so the growth factor binds, the RTKs pair up, dimerize, and they phosphorylate each other, right? Autophosphorylation.
Exactly, and those new phosphate groups act like little docking sites, little sticky pads for other proteins to bind to.
And this is where adapter proteins jump in, like GRB2, I remember that one, GRB2.
That's a key one, yeah.
Yeah.
GRB2 docks onto the phosphorylated RTK. And GRB2 then grabs another protein called SOS.
There's OS.
And an SOS is really important here. It's a guanine nucleotide exchange factor, a GEF.
Right, GEF. So signal comes in, hits the RTK, GRB2 links it to SOS. What's SOS doing?
SOS interacts with the star of this pathway.
RAS.
RAS proteins, there are a few. HRAS, KRAS, NRAS are these small GT passes. Think of them like molecular switches.
Right.
They live near the cell membrane.
Right, and they're off when they're bound to GDP, right? Guanine diphosphate.
Correct. GDP-bound RAS is inactive. SOS, the GEF, comes along and facilitates RAS dropping the GDP and picking up a GTP guanine triphosphate.
Ah, so SOS promotes the swap, GDP out, GTP in, and now RAS is active, RAS GDP.
Exactly. RAS GTP is the on switch. Now, RAS does have a way to turn itself off. It can slowly hydrolyze that GTP back to GTP, but this is usually accelerated by another set of proteins called GTPase activating proteins, or GCAS. Like NF1 is a well-known GTP.
Okay, GIPs speed up the off switch, but this is where the cancer link comes in, right? Those RAS mutations.
This is huge. Oncogenic mutations in RAS genes, often at codons 12, 13, or 61, basically jam the switch. They trap RAS in that active GTP-bound state.
So it can't turn itself off properly, even with G-gaps around.
Right. It's just constantly signaling grow, 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 even if there are no growth factors telling it to. And these mutations are found in, something like 30% of all human cancers. It's massive.
Wow. Okay, so RAS stuck on, what happens next downstream?
Active RAS GTP then kicks off this really important kinase cascade, RAF, then MEK, then ERK.
RAF, MEK, ERK, RME. That mnemonic definitely helps keep them straight.
It does. So active RAS recruits and activates the RAF kinases ARAF, BRAF, CRAF. BRAF is another one often mutated in cancer, like melanoma.
Right. So activated RAF then phosphorylates MEK.
Yep. RAF phosphorylates and activates MEK1 and MEK2. MEK1-2.
And then MEK phosphorylates ERK, ERK12, also called P42, P44, MEP kinase.
Precisely. That's the MEP kinase right there.
Yeah.
MEK activates ERK.
Okay, so now ERK is active. What does it do?
Activated ERK actually moves, it translocates into the nucleus, and inside the nucleus it finds and phosphorylates transcription factors. Key ones are ELK1, and components of the AP1 complex like CFOs and C-gene.
And these transcription factors are the guys that actually turn on the genes?
Exactly. They bind to DNA and switch on genes needed for the cell cycle to progress, especially things like cyclin D1. This helps push the cell through that critical G1S checkpoint in the cell cycle, basically telling it to divide.
So summarizing that chain, growth factor signal, RTK RES switch, stuck on in cancer. The RME cascade, RAF-MEK ERK goes nuclear, activates transcription factors, drives proliferation.
You got it. That's the core pathway.
Okay, clear. Let's switch to the second big one, PI3K mTOR. PI3K mTOR. If RAS-MEK is the gas pedal for proliferation, this one feels more like survival and growth control, and metabolism.
That's a great way to think about it. It's a master regulator of all those things. Cell growth, survival, anti-apoptosis, and metabolism. And yeah, it's often initiated by the same RTKs we just talked about.
Or even by active RAS itself, right? They're interconnected.
Definitely interconnected. Active RTKs, or active RAS GTP, can recruit and activate PI3 kinase, PI3K. PI3K itself is usually made of two parts, a regulatory subunit, often P85, and a catalytic subunit, P110.
Okay, so PI3K gets activated. What's its main job? It's a kinase, but it targets lipids.
Exactly. That's the key difference here. PI3K is a lipid kinase. Its critical job is to phosphorylate a lipid molecule found in the cell membrane called PIP2, PIP2. That's phosphatidylinositol 4-ferricyl-5 bisphosphate.
Okay.
PI3K adds a phosphate group to PIP2, turning it into PIP3, PIP3, phosphatidylinositol 3-fevlin-4-carbyl-5 tris phosphate.
So PIP3 is the crucial product here, a lipid second messenger.
Yes, PIP3 is super important. It stays in the membrane, but its presence creates docking sites, like little landing pads, for proteins that have a specific domain called a pH domain.
And AKT has one of those pH domains. That's how it gets involved.
Exactly. The buildup of PIP3 recruits AKT, also called protein kinase B, or PKB, to the membrane. It also recruits another kinase called PDK1.
So they both come to the membrane because of PIP3.
Right, and having them both there, co-localized, is critical. PDK1 can now phosphorylate AKT at a specific spot, 3-non-308. 3-non-308. Is that enough to activate AKT? Almost. It needs one more phosphorylation event for full activation. This second hit comes from a complex called mTORC2. That's mTOR complex 2, mTORK2. mTORC2 phosphorylates AKT at serine 473.
Ah, okay. So PDK1 at 308 and mTORC2 at SIR 473. 2 hits for full power.
You got it. And once fully active, AKT is a central hub. It phosphorylates many downstream targets to promote those key outcomes: survival, growth, proliferation, and metabolism.
How does it boost survival, keep cells from dying?
It does that mainly by blocking apoptosis. It phosphorylates and inactivates pro-apoptotic proteins. Examples are BAD, BAD, and caspase-9. It can also activate MDM2, which targets the tumor suppressor P53 for degradation.
Getting rid of P53, that's a major survival signal.
Huge. And AKT also activates NF-kappa B, and F-kappa B, another transcription factor that turns on pro-survival genes. So multiple ways AKT is basically shouting, don't die, at the cell.
Okay, and what about the growth side? How does it connect to mTOR, specifically mTORC1?
Right, so AKT activates mTOR complex 1, mTORC1. One key way it does this involves a complex called TSC1-TSC2. Normally, TSC2 acts as a GP for a small GTPase called REB, keeping REB inactive.
Okay, another little switch protein like RAS.
Exactly. AKT phosphorylates and inactivates TSC2. When TSC2 is inactive, it can no longer inhibit REB, so REB becomes active, REB GTP, and active REB directly binds to and activates mTORC1.
So AKT knocks out the inhibitor, TSC2, allowing the activator, REB, to turn on mTORC1.
Precisely. And active mTORC1 is a major driver of cell growth. It promotes protein synthesis by phosphorylating targets like S6 kinase, S6K, and 4EBP1. This basically ramps up the machinery for making proteins, which is essential for cells to grow bigger and divide.
Turning up the factory's production lines.
Exactly. Now, just like RAS-MAPK had GPs, this pathway needs a break too. And the critical break here is PTEN.
Yeah.
That's a tumor suppressor, right?
A major one. PTN is a lipid phosphatase. It does the opposite of PI3K. Ah, so it removes the phosphate from PIP3, turns it back into PIP2.
Precisely. PI3 phosphorylates PIP3 back to PIP2. By removing the PIP3 signal, it shuts down AKT activation and everything downstream. It's the off switch for this pathway.
And losing PTEN function, which happens a lot in cancers like prostate and endometrial cancer?
It means the break is gone. You get constitutive, non-stop signaling through PI3K, AKT, mTOR because PIP3 levels stay high. This is why it's one of the most commonly hyperactivated pathways across many cancers.
That makes total sense. No break, pathway runs wild.
Right. Let's try that factory analogy again to really cement it. Think of the cell as this big factory. Needs production, growth, security, survival, energy, metabolism. PI3K, AKT, mTOR is the central control room. Orders come in, growth factors rose. PI3K is the dispatcher sending out the key memo, PIP3, across the factory floor. That memo, PIP3, attracts the key managers, AKT being the main one. AKT gets signed off by its supervisors, PTK1, mTOR, C2, and becomes the general manager. It starts shouting orders. Ramp up production via mTOR, C1. Beef up security, no shutdowns. Inhibiting bad caspases, activating NFKB, get more power, metabolism.
Makes sense.
And PTN. PTN is the crucial safety inspector. Their job is to constantly monitor that PIP3 memo level. If things get out of control, PTN shreds the memo, dephosphorylates PIP3, hitting the reset button.
But if the inspector, PTNN, is gone or mutated?
The control room is stuck on full blast. Unchecked growth, survival, everything. The factory just expands uncontrollably and resists being shut down. That's what happens with the PTN loss.
Okay, that factory analogy definitely helps visualize PTN's role as a brake. It really clicks now.
Good. Okay, pathway number 3, HIF-1α, HIF-1α. This one's different. It's not primarily about growth factors, it's about the cell's oxygen level, specifically low oxygen or hypoxia.
Right. Tumors often grow faster than their blood supply can keep up, leading to these hypoxic zones. Cells trapped there need to adapt or die.
Exactly. And the hypoxia-inducible factors, HIFs, are the main system for sensing and responding to low oxygen. HIF-1 is the key player we focus on.
And HIF-1 is actually two proteins working together.
Yes, it's a dimer. It consists of a subunit that's always around called HIF-1 beta, or RNT, or NT, and the really important oxygen-regulated subunit, HIF-1 alpha. The whole game is about controlling the level of HIF-1 alpha protein.
So HIF-1 alpha stability is the key control point.
That's it. Under normal oxygen conditions, normoxia of the cell actively destroys HIF-1 alpha to keep its levels low.
How does it do that? It involves adding hydroxyl groups, right?
Correct. Specific enzymes called prolyl hydroxylase domain proteins, or PHDs, use oxygen as a substrate to add hydroxyl groups, or H, onto specific proline residues within the HIF-1 alpha protein.
Okay, so PHDs need oxygen to work. Makes sense. They hydroxylate HIF-1 alpha, then what?
That hydroxylated HIF-1 alpha is now recognized by another crucial protein, PVHL. It's the von Hippel-Lindau tumor suppressor protein. PVHL is part of a larger complex that functions as an E3 ubiquitin ligase.
Ubiquitin ligase, so it tags HIF-1 alpha for the garbage disposal.
Exactly. PVHL puts ubiquitin tags, UBIK on hydroxylated HIF-1 alpha. Ubiquitin is the signal for the proteasome, the cell's protein degradation machinery, to come and chew it up.
So normoxia, oxygen available, PHD's active HIF-1 alpha hydroxylated VHL binds ubiquitination destruction. Levels stay low.
Perfect summary. There's also another oxygen-dependent enzyme, FIH1, that hydroxylates an asparagine residue on HIF-1 alpha in normoxia, which prevents it from recruiting transcriptional coactivators, another layer of off signal.
Okay, but what happens when the oxygen level drops? Hypoxia.
Here's the switch. In hypoxia, there isn't enough oxygen for the PHDs and FIH1 to work efficiently.
So no hydroxylation.
Or much less of it. And if HIF-1 alpha isn't hydroxylated on those key prolines, PVHL can't recognize it.
No VHL binding means no ubiquitination.
And no proteasomal degradation. Yep. So HIF-1 alpha, which is constantly being made, is no longer being destroyed. It rapidly accumulates in the cell. It stabilizes.
Got it. It builds up. Then what does stabilized HIF-1 alpha do?
It travels into the nucleus, finds its partner HIF-1 beta, RRNT, and they form the active HIF-1 transcription factor dimer. This dimer then binds to specific DNA sequences called hypoxia response elements, HREs, located in the promoter regions of target genes.
And these target genes help the cell cope with the low oxygen.
Precisely. It switches on a whole survival program. Key targets include VEGBBF to promote angiogenesis, trying to grow new blood vessels to get more oxygen, enzymes involved in anaerobic glycolysis like GLUT1, GLUT1 to bring in more glucose, and BDK1, different from the AKT activating BDK1, to shift metabolism away from oxygen-requiring pathways. This is the Warburg effect in action. Also EPO, and genes involved in cell survival, invasion, and pH regulation.
It's like the cell flipping a master switch to go into survival mode in a hostile low-oxygen place.
Absolutely. And just like PPN loss affects the PI3K pathway, mutations that inactivate VHL, as seen in von Hippel-Lindau disease and many sporadic clear cell renal cancers, cause HIF-1α and HIF-2α to be stable even when oxygen is normal. The degradation system is broken, so the hypoxia response is always on.
Wow. Okay, so we see how these pathways work normally or how they get broken. Let's really focus now on why this matters so much in oncology, especially for us in radiation oncology.
It's fundamental because these pathways, when dysregulated, are driving the cancer phenotypes we struggle against. Constitutive RAS MAPK, uncontrolled proliferation, hyperactive PI3K mTOR, increased survival, resistance to apoptosis, altered metabolism, stabilized HIF-1α, adaptation to hypoxia, angiogenesis, metastasis potential. They hit multiple cancer hallmarks.
And figuring out which pathway is driving a particular tumor is getting more and more important for treatment decisions.
Exactly. That's where molecular diagnostics come in. Sequencing tumor DNA to look for those mutations, KRAS, BRAF, PIK3CA, PIK3CI, encoding PI3K's P110 subunit, PTN loss, VHL mutations, or using immunohistochemistry, immunohistochem on the biopsy to actually see if proteins like phosphorylated AKT or HIF-1α are abundant.
Right, connecting the dots back to the path report.
Yeah, this information helps predict how aggressive the tumor might be and, crucially, whether it might respond to specific targeted drugs.
Okay, let's zero in on radioresistance. How do these pathways make our radiation treatments less effective?
They're huge contributors. Take PI3K and KT. Its main job is survival, right? Blocking apoptosis. Well, radiation works partly by inducing apoptosis. If AKT is hyperactive, it's constantly putting up shields, making those cells much harder to kill with radiation-induced damage. And FKB activation helps too.
So we hit them with radiation, damage their DNA, but the cell's internal signaling just says, nah, we're good, don't die.
Pretty much. Then you have RAS MAPK driving that constant proliferation. Remember accelerated repopulation. If the surviving tumor cells divide really fast between radiation fractions, the tumor can essentially regrow, undermining the treatment effect.
The tumor is just outrunning our daily treatment.
Exactly. And then there's hypoxia, orchestrated by HIF-1α. We know the oxygen effect is critical for radiation sensitivity. Oxygen helps fix the DNA damage caused by radiation-free radicals, making it permanent and lethal. Hypoxic cells lack that oxygen.
So they need way more dose, like 2.5 to 3 times more, for the same kill.
Right. But it's worse than just the oxygen effect. HIF-1α itself, when stabilized in hypoxia, turns on genes that also promote survival, sometimes including DNA repair genes and anti-apoptotic factors. It adds extra layers of resistance specific to those hypoxic cells.
And didn't you mention ACT might also directly affect DNA repair?
Yeah, there's growing evidence suggesting ACT can influence the efficiency of DNA double-strand break repair, possibly making cells better at fixing the very lesions radiation causes.
So multiple hits, pro-survival signals, rapid regrowth, resistance due to low oxygen, and potentially even enhanced DNA repair. It's the perfect storm for radioresistance.
It really is. And that's precisely why these pathways are such attractive targets for drug development, especially in combination with radiation.
Right. Let's talk about those drugs. We have agents hitting the start of the pathways, the RTKs.
Yep. EGFR inhibitors are a big class. Monoclonal antibodies like cetuximab, cetuximab, and panitumumab, and the small molecule tyrosine kinase inhibitors, TKIs, like gefitinib, erlotinib, afatinib, osimertinib, osimertinib, especially important in lung cancer with specific EGFR mutations.
And moving down the RAS MAPK chain.
We have RAF inhibitors like vemurafenib, vemurafenib, and dabrafenib, mainly for BRAF mutant melanoma, and MYC inhibitors like trametinib, and cobimetinib, often used together with the RAF inhibitors to block the pathway more effectively.
And the direct RAS inhibitors are finally here.
Yes, it's really exciting. Drugs like sotorasib, and adagrasib that specifically target the KRAS G12C mutation are now approved for certain cancers, like lung and colorectal. A huge breakthrough after decades of trying.
Okay, what about targeting PI3K mTOR?
Lots of action there, too. We have PI3K inhibitors, some specific to certain isoforms like idelalisib for blood cancers, or alpelisib for PI3K mutant breast cancer. AKT inhibitors are in trials, like capivasertib, and mTOR inhibitors, the rapalogs, Rapologs like everolimus and temsirolimus have been around for a while for certain cancers like renal cell and neuroendocrine tumors.
And hitting the hypoxia response, mostly anti-angiogenesis.
Primarily, yeah. Targeting VEGF, the main output of HIF for blood vessels. The antibody bevacizumab targets VEGS itself. And then there are VEGFR TKIs drugs that block the VDEF receptor like sunitinib, sunitinib, sorafenib, sorafenib, pazopanib, used in kidney cancer, liver cancer, sarcomas, etc. The idea is to choke off the tumor's blood supply or maybe normalize the vessels to improve drug and oxygen delivery.
And the big hope is combining these targeted agents with radiation to overcome resistance, right? Like using EGFR inhibitors with radiation for head and neck cancer.
Exactly. That's a major focus of clinical trials, finding the right drug for the right patient based on their tumor's pathway alterations, combined optimally with radiation.
Okay, let's try that mini case idea again. Imagine that pancreatic cancer patient we know, say, 90% have activating KRAS mutations.
Okay, KRAS mutant pancreas cancer, what does that immediately tell you?
Well, RAS-MAPK pathway is likely driving proliferation hard, probably predicts aggressive behavior, rapid growth.
Yep. So radiobiologically, what concern does that raise?
Accelerated repopulation between fractions makes timely treatment delivery really important.
Good. Now, if the main driver is that mutated KRAS, what about using an upstream inhibitor, like an EGFR antibody?
Probably not going to work well, right? The signal's already locked on downstream at RAS. Blocking the receptor won't shut off the pathway.
Exactly. Which matches the clinical data showing KRAS mutations predict resistance to EGFR antibodies in colorectal and pancreatic cancer. So understanding that pathway helps predict drug response, or lack thereof.
And highlights why we need drugs that can hit mutated RAS or things downstream like Merck if we want to combine them effectively with RT in these tumors.
Perfect. See how the basic science informs clinical strategy?
Definitely. That really clarifies the connection.
All right, let's nail down some clinical pearls. High-yield stuff. Pearl number 1.
Mutational status matters. Knowing if KRAS, BRAF, PIK3CA are mutated, or if PTN is lost, is key for predicting response to targeted agents, especially EGFR inhibitors. KRAS mutation, likely resistance to cetuximab panitumumab in colorectal cancer.
Pearl number 2. Hypoxia equals radioresistance, primarily due to the oxygen effect, and HIF-1α is the master regulator of that hypoxic response. Remember that 2.53x dose requirement.
Pearl number 3. PI3K is the big pro-survival pathway. Activation makes cells inherently resistant to radiation-induced apoptosis. It's like a shield against cell death.
Pearl number 4. Targeting these pathways can potentially radiosensitize, yes, but combining targeted drugs and radiation is tricky. Interactions can be complex, affecting tumor cells, blood vessels, oxygenation. Figuring out the best combinations and timing is still a work in progress.
Excellent pearls, very board relevant.
Mnemonic segment note. Insert a mnemonic here if one was developed for the RAS-MAPK cascade or another pathway component.
Okay, ready for a quick board blitz? Let's test this. Question 1.
The RAS protein cycles between active GTP-bound and inactive GDP-bound states. What activates RAS by promoting the GTP to GTP exchange? A. GAPs, GTPase activating proteins. B. PKA, protein kinase A. C. GEFs, guanine nucleotide exchange factors like SOSD, inositol transferase.
Okay, focus on the exchange GDP out, GTP in. What helps that happen?
The answer is C. GEFs, like SOS. Guanine nucleotide exchange factors promote the swap. GKs do the opposite. They help RAS hydrolyze GTP back to GDP, turning it off. Farnesyl transferase just anchors RAS to the membrane.
Question 2.
Which tumor suppressor acts as a lipid phosphatase to directly oppose PI3K KT signaling? A. P53, B. RBC, PTN, D. VHL.
Remember the brake pedal for the PI3K pathway, the one that reverses PI3K's action?
Correct answer is C. PT10. PT10 removes the phosphate from PIP3, turning it back to PIP2, thus shutting down AKT signaling. P53 and RB are cell cycle apoptosis regulators. VHL targets HIF-1α.
Question 3.
Under normal oxygen, or hypoxia, how are HIF-1α protein levels kept low? Less HIF-1α gene transcription. B. Phosphorylation by ATM. C. Hydroxylation by PHDs, then VHL-mediated ubiquitination and degradation. Desequestration by IKB.
Think about the oxygen-dependent step that marks HIF-1α for destruction.
The answer is C. Hydroxylation by PHDs, followed by VHL recognition, ubiquitination, and proteasomal degradation. This whole chain depends on oxygen for the initial PHD step.
Last one for the blitz.
A tumor has constitutive activation of the RAF-MEK pathway. Which drug class most directly targets this cascade? A. Anti-VEGF antibody, bevacizumab. B. PRP inhibitor, olaparib. C. MEK inhibitor, trametinib. D. Aromatase inhibitor, letrozole.
Look for the drug that hits one of the kinases within that specific RAF-MEK
The answer is C. A MEK inhibitor like trametinib. It directly inhibits MEK, a core component. Bevacizumab hits VD angiogenesis. PRP inhibitors hit DNA repair. Aromatase inhibitors hit estrogen synthesis. You want the direct pathway inhibitor here.
Nicely done. Getting those mechanisms and key players straight is crucial.
Definitely. Okay, so we've got the basics, the drugs, the resistance, but it's not static, right? This field is always moving.
For sure. And combining these targeted agents with RT isn't straightforward. You mentioned the anti-angiogenic timing dilemma. Normalize vessels first, or suppress them after. Big questions remain.
Absolutely. And resistance is a huge issue. Tumors are clever. They find ways around these drugs. They might upregulate a parallel pathway or gain new mutations in the target itself or downstream.
So the research focus now is really on understanding those resistance mechanisms, developing next-gen drugs, maybe hitting multiple targets, and figuring out the smartest combinations and sequences with radiation and other therapies.
Exactly. It's all about trying to stay one step ahead of the tumor's adaptability.
Okay, let's try to pull this all together as we wrap up not just this session, but the whole RadBio series.
We covered 3 massive signaling pathways today. First, RAS-MAPK, the proliferation engine. RAS is the GTP-GDP switch activated by RTKs, firing off the RAF-MEK cascade to drive cell division. Remember RME.
Then PI3K AKT mTOR, the survival growth and metabolism coordinator, also kicked off by RTKs or RAS. PI3K makes PIP3 activating AKT, and PTN is the critical off switch, the lipid phosphatase brake, remember the factory analogy?
And third, HIF-1α, the hypoxia response master. HIF-1α protein level is the key. Stable in low oxygen because PhDs can't hydroxylate it, leading to VHL escape. It then turns on genes like VEGF and GLUT1 for adaptation.
And the big picture, when these pathways are broken in cancer, mutated RAS, lost PTNN, hyperactive AKT, stable HIF, they fuel tumor growth, survival, metastasis, and critically for us, make tumors resistant to radiation.
Which is why targeting them with drugs, EGFR inhibitors, MEK inhibitors, PI3K mTOR inhibitors, anti-VEGF agents is such a key strategy, especially finding ways to combine them effectively with radiation to overcome that resistance.
Pure. And with that, that's a wrap on our radiation biology series.
We actually did it. Wow. From the basic physics, photons, electrons, DNA damage and repair, cell cycle, cell death modes, tumor microenvironment, normal tissue effects, all the way to these complex signaling pathways today. We covered a ton of ground.
We really hope this series has helped build a solid foundation for you, not just for acing your boards, but for really understanding the why behind what we see in the clinic. This molecular stuff is becoming more integrated every day.
Yeah, and remember, the key is review, review, review. Don't feel like you have to absorb it all at once. It's a lot. Chip away at it. Maybe don't try to memorize the whole textbook overnight, you know?
Yeah, definitely not. Maybe just focus on, not getting hypoxia yourself while studying.
Or just remember the oxygen effect. Sometimes the simplest concepts tie everything together. It's proof that, yeah, sometimes the basics are the most profound.
Seriously, though, it's been fantastic going through all this with you all. Thanks for sticking with us through this whole RadBio adventure with Rad Onc Smart Review.
It's been fun. Remember, you can find practice oral boards and other resources over at radoncsmartlearn.com. And subscribe to Rad Onc Smart Review for whatever craziness we tackle next.
Until then, keep learning, keep questioning, and good luck with everything, especially those upcoming exams.
Thanks so much for tuning in.